A very good afternoon to everyone. Today we will study about plasma membrane and the transport through plasma membrane. We all know that plasma membrane is selectively permeable. It only allows fewer molecules to pass over it, and it acts as a barrier so that the molecules cannot interact directly to the cytoplasm to the outer environment. In human RBC, 40% are lipid and 52% are protein, forming the plasma membrane. The plasma membrane transport molecules via two methods. That the first one is the passive method and the second is the active transport. The first one is the simple method of transport of molecules via it, that only depends on the concentration gradient. In some cases, it depends on the electrochemical gradient for the polar molecule. But this passive transport is of two types: simple diffusion and facilitated diffusion. Simple diffusion is a concentration gradient diffusion where the molecules passes over from one side to the another side of the plasma membrane, and it includes the water molecule and neutron molecule, that is oxygen, CO2 and benzene. While through facilitated diffusion, the passage of molecules across the plasma membrane occurs via transporters that may be a carrier or a channel. The carrier molecule includes the example of glucose transporter that help in the transport of glucose via plasma membrane. The channel includes the aquaporine, that is a special protein or the channel molecules for the transport of water via plasma membrane. Now the second type is the active transport, where the transport of molecule is against the concentration gradient and that occurs by carrier proteins. This transport of molecules is always from downhill to uphill, means the concentration is lower but the movement of particles occur to the higher concentration, and that meets the energy source. But this active transport is further of two types: primary active transport and secondary active transport. The primary active transport is the transport where the molecules that transported from one side of the plasma membrane to the other side is directly coupled to the energy source, that is to the ATP hydrolysis, and the example include the sodium and potassium pump. While the secondary transport is the active transport type, where the molecule is not directly attached to the energy source, where it is attached to other molecule, and that together they are transported in same direction or other direction, and the energy is utilized. This secondary active transport is of two types: symport and antiport. If the two molecules are moving in the same direction and the energy is utilized, then it is called as the symport secondary active transport, and the example is sodium glucose pump that is present in the small intestine. The second is the antiport secondary active transport, where the two molecules move opposite to each other and the energy is utilized, and the example includes the lactose permease, where the proton and lactose are transported oppositely, and that is present in the E. coli bacteria. Now, 2 or 3 points that has to be added with the cell membrane and the cell wall. Now, cell wall is present in fungi and plants, and it's a non-living part of a cell. It is important for the shape and protection of a cell from the outside environment. This cell wall of algae is made up of cellulose, galactans, mannans and calcium carbonate, while the cell wall that is present in the plants is made up of cellulose, hemicellulose, pectins and proteins. Further, there is a middle lamella that is present in the plants that is made up of calcium pectate and glue neighboring cells. There is also a word that is called plasmodesmata, and that is a part of plants that is present or transverses the cell wall and the middle lamella. Now we will study about important system present in a cell that is called as the endomembrane system. This endomembrane system is present in a cell and include the membranous structures present inside it. These membranous structures work in a coordinated order and include endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. First we will study about the endoplasmic reticulum. Endoplasmic reticulum is the tiny reticular structures or network of tubular structures present in continuation to the nuclear membrane, and it has two compartments, the luminal side having the lumen and that is inside the endoplasmic reticulum, while the outer part is called as the extraluminal and it is present inside the cytoplasm. This is the simple figure showing the reticular structure of the endoplasmic reticulum, which is in continuation with the nucleus, and these are the small molecules or the ribosomes present outside the surface of the endoplasmic reticulum, and this is called as the RER, rough endoplasmic reticulum. While if ribosomes are absent on the reticular structure of the endoplasmic reticulum, then it is called as the SER, smooth endoplasmic reticulum. Thus, endoplasmic reticulum is of two types, rough ER having the ribosomes, and these are involved in protein synthesis and secretion, while the endoplasmic reticulum not having the ribosomes or ribosomes are absent on their surface are involved in lipid synthesis. Now, the second endomembranous system is Golgi apparatus. It was discovered by Camillo Golgi in 1898. It's a flat, disc-shaped cistern and its size varies from 0.5 to 1 micrometer in diameter. It's involved in the packaging of materials for transport inside the cell or outside the cell. And also it's involved in the formation of glycoprotein and glycolipids for plasma membrane. Golgi is a disc-shaped cisternal structure and it has two phases, the cis phase and the trans phase. Cis phase is having the convex phase and trans phase is having the concave phase. The cis phase is convex and it is near to the nucleus, while the trans phase is a concave phase and it's called the maturing phase. The most important point is that the Golgi apparatus is involved in the transport and packaging, where the molecules or the particles which has to be packaged in the form of vesicles from endoplasmic reticulum merges to the cis phase of the Golgi and they move from one cisternae to another and get encapsulated, and then they move out through its maturing phase, and then they are moved outside the Golgi into the cytoplasm if they are required there, or outside the cell where they have to be sent. Now, two more organelles, lysosome and vacuoles, are the part of endomembranous system. First, the lysosome is the organelle that is rich in hydrolytic enzymes that hydrolyzes the particles and the molecules, and it has a number of hydrolases that include lipases for the digestion of lipids, proteases for the digestion of proteins, and carbohydrates for the digestion of carbohydrate. It's always active at the acidic pH and it's formed during the process of packaging of materials from Golgi apparatus. The last one is the vacuoles. These are membrane-bound spacious structures are involved in the storage of water, sap or excretory products. In plants, majority of the plants have 90% of the vacuoles, and these are involved in the facilitated transport of ions in the plant cells. We have two important examples for vacuoles where the amoeba an organism has a specific contractile vacuole that is involved in the excretion process, while in the protista they have a large food vacuole to engulf the food. Thus, we have studied 4 parts of an endomembranous system. The first one was the endoplasmic reticulum, second was the Golgi apparatus, third one is the lysosome, and the fourth is the vacuole. These all 4 membranous structures are together, work in a coordinated fashion, and are very important for the cellular functioning of a cell.